In this lecture, I will talk about class II MHC molecule and how antigens are processed and presented by this molecule. But before beginning, I want to mention some important points about the class II MHC molecule. The first important point is this class II MHC molecule is actually expressed by very few cells like professional antigen presenting cell like macrophage, dendritic cell and B cell, and thymic epithelial cell and endothelial cell. This class II MHC molecule is actually recognized by a subset of T cell called T helper cell, that is also known as CD4 positive, because this helper cell or this subset of T cell contain a co-receptor called CD4 positive. This T helper cell actually express T cell receptor that recognize the class II MHC molecule. The second point is dendritic cell express MHC2 molecule constitutively. It means dendritic cells express all the time class II MHC molecule. B cells also express class II MHC molecule constitutively, but macrophage do not express MHC2 molecules constitutively. It means when macrophage get activated, only it express class II MHC molecule. The next point is the class II MHC molecule contain two polypeptide chain. One is alpha chain and another is beta chain. So let me draw a class II MHC molecule. This is a nucleus. If this is a dendritic cell or macrophage or B cell, then this cell can actually present or express class II MHC molecule. Class II MHC molecule looks like this. If this is the alpha chain, then this is called alpha 1 domain and alpha 2 domain, and if this is a beta chain, this is called beta 1 domain and beta 2 domain. Now this alpha 1 and the beta 1, this region, this domain actually, alpha 1 domain and beta 1 domain actually make peptide binding cleft or groove. So a peptide can bind in this region. Okay. Another point you have to remember in case of class II MHC molecule, this alpha 1 and beta 1 domain show polymorphism. In my previous class, I have already mentioned that this MHC molecule is polymorphic in nature. But in case of class II MHC molecule, all the domain of the molecule is not polymorphic. Only the peptide binding region of the MHC2 molecule is polymorphic. Okay, this is a very important point. And another point you have to remember, the size of the peptide that can bind with the peptide binding cleft. Then the size is, the range is 13 amino acid to 18 amino acid, or it could be larger. In case of class I MHC molecule, I have mentioned that only 8 to 10 amino acid peptide can bind in the peptide binding cleft or groove. But in this case, class II MHC molecule can accommodate a larger size of peptide molecule. And the last point you have to remember, this is the most important point of the class II MHC molecule because it is related with the antigen processing and presentation. The way by which the class II MHC molecule actually present the antigen is called exogenous pathway. So what is exogenous pathway? Exogenous means outside of the cell. So when pathogen or bacteria is coming from the outside, and not only that, and the antigen processing is occurring in a vesicle, not in the cytoplasm. In case of class I MHC molecule, the antigen are processed, means cleaved or chopped in the cytosol by immunoproteasome. But in case of class II MHC molecule, the antigen are processed into a vesicles. Like this class II MHC molecule actually represent or present this exogenous antigen, and the source of exogenous antigen by phagocytosis. The pathogen enter into a vesicle called phagosome. Like when phagocytosis occur, like dendritic cell and macrophage can perform phagocytosis. Now the pathogen enter into the phagosome. The second way by which antigen are processed, receptor-mediated endocytosis. I am just writing the endocytosis, but receptor-mediated endocytosis. B cell perform this function, receptor-mediated endocytosis. Dendritic cell and macrophage cannot perform this endocytosis, this type of endocytosis. So what happens when antigen or bacteria are engulfed by receptor-mediated endocytosis? The pathogen or antigen is present in a vesicle called endocytic vesicles. And the last point is autophagy. So this autophagy, when performed, the self-antigen or the cell's organelle are present in the autophagosome. This autophagosome is also a vesicle. So these all are vesicles. Okay. So these are the all basic facts about the class 2 MHC molecule.